There are cellular components. Here we go. You have the extracellular matrix, which is on the outside of the cell, but connected to the inside by the cytoskeleton. In animal cells, it's secreted by the cell and it fills gaps between individual cells. It is a mixture of protein fibers, which can be collagen and elastin, embedded in hydrated matrix of glycoproteins. The functions are adhesion between cells, providing structure, influencing cell behavior, and links to the cytoskeleton inside the cell. The plasma membrane. This surrounds the cell, acts as a barrier, and dictates what enters and leaves the cell. It is composed of phospholipids, lipids, and membrane proteins, and forms a biolayer with proteins suspended in it, and these are usually used as transporters. Carbohydrates are often on the outside and they stick out like this, and they are used for recognition, so the cell can recognise it as itself, as the correct hormones can attach to them. The glycocalyx is used in the smooth passage of food and protects the gut from enzymes. It is a carbon-rich zone, and it protrudes from the outside of the surface of the cell membrane, which can be bound to proteins and lipids. It's involved in recognition as well as adhesion and protection of the cell surface. There are two types attached, which is the carbohydrate, and it's firmly bound to the proteins and the lipids for recognition usually. There's unattached as well, which is easily removed, and these are the intestines, and they're secreted by the cell. Oh no, these secrete mucus. There is the cytoplasm, the cytosol, and the cytoskeleton. The cytoplasm is everything in the cell, all organelles, everything. The cytosol is a semi-fluid liquid thing, which everything floats in, so the organelles and everything suspended in that. The cytoskeleton is composed of a 3D network of interconnecting filaments and tubules. It has a role in the movement of the organelles, movement of the cell itself, movement of chromosomes when they divide, and also maintaining the cell shape. Then we have the nucleus. Its boundary consists of two membranes which have pores in it, so water and ribosomes can come in and out, and proteins as well. The nucleoplasm contains the chromosomes when not in use, when not dividing, and they are dispersed as DNA proteins called chromatin. The nucleus synthesizes the RNA necessary for ribosomes. There is the endoplasmic reticulum, and there's a smooth one, which is tube-shaped, cristae, involved in fatty acid, lipid, and steroid synthesis, as well as carbohydrate metabolism. It is also the source of calcium storage. Rough one is flattened cristae dotted with ribosomes, and is used in protein synthesis, and occurs at the ribosomes and glycosylation of the proteins. The Golgi apparatus. It looks like a stack of plates and is the main organelle for sorting, processing, and secreting proteins. There are usually 3 to 8 cristae per stack, and there can be one or there can be lots of Golgi in the cell. Lysosomes are 0.5 to 1 micrometer in length and stuff. And the organelle, which stores the fatty acid hydrolases, and they break down all cellular components and molecules that are taken up from the outside of the cell. Mitochondria are the size of a bacterial cell. Aerobic respiration takes place to extract energy, converting glucose into ATP. Tricarboxylic acid cycle occurs inside the mitochondria, which has its own DNA. The membranes compartments all separate reactions to occur, so if they weren't separate they couldn't occur, because they're different enzymes and they need different environments to work. The peroxisomes are similar size to the lysosomes and are found in most cells, but they are prominent in liver and kidney cells. They play a role in the detoxification of hydrogen peroxide by catalase. Also, they are involved in the breakdown of long fatty acids. Ribosomes have no membrane. In eukaryotic cells are thousands, because like 25 to 30 nanometers, so they're really tiny. And ribosomes are composed of two subunits and are the site of protein synthesis.